Welcome to day 2. At this point, we have isolated the DNA, broken it into random 3 kb pieces, inserted the pieces into the vector, and forced those plasmids into E. coli bacteria. The bacterial colonies have had about 20 hours to grow on the medium containing ampicillin and X-gal. The E. coli bacteria divide about every 20 minutes. So in 20 hours, we have about 1 million bacteria per colony. On this plate, we have 3 different kinds of bacteria. The first is the bacteria that did not receive a plasmid. You can't see them because they are dead. That's 99.9% of the transformation stock that did not receive a plasmid. They did not get the gene for antibiotic resistance from the plasmids. The second kind is the blue ones here. They received a plasmid and the gene for ampicillin resistance. However, if you remember, certain plasmids did not incorporate a sheared segment of DNA in the middle of the LAC-Z gene. So these blue colonies have the PUC18 plasmid, but the plasmid has a functioning LAC-Z gene, which means that they do not contain the DNA we want. Finally, we have these white colonies. These colonies received a plasmid, and that plasmid had a fragment of the genomic DNA inserted in the LAC-Z gene, which disrupts its function. All of these white colonies together more or less contain the sum of the original long 150 to 200 kilobase source strand of DNA we started with. All these plates that contain the same genome are together called a library. Each of the white bacterial colonies arose from a single bacterium, which took a single plasmid with a single clone fragment of DNA. So we have effectively separated each fragmented segment of the original DNA strand and replicated it 1 million times. And we can actually see it right here. The next step is to transfer these E. coli cells on this bioassay to a 384-well microtiter plate. This plate contains liquid medium, which promotes the growth of the bacterial cells. This plate has also allowed us to automate this process from this step forward. The QPix machine here uses an imaging system to identify and pick the white colonies from each agar plate and place them in a well of these microtiter plates. At this point, the plates are placed into the incubator to grow for another 18 to 20 hours. Here we are on day 3 of the process. So far, we've collected the DNA, fragmented it into 3 kilobase segments, inserted those segments into the plasmids, and inserted the plasmids into the E. coli bacteria. We let the bacterial colonies of E. coli grow and transferred the colonies containing the cloned fragments of genomic DNA into these 384 well plates. A single well on these plates contains a colony of E. coli bacteria. Each bacteria contains a plasmid with our DNA insert that we want to sequence. Each well contains a different genomic DNA clone. We are almost ready to read the DNA sequences. But first, we have to crack open the E. coli bacteria and get out the DNA we want to sequence. The first step is to place the 384 well plates into a PlateMate Plus robot. This transfers the E. coli into a new 384 well plate that contains a TE buffer solution, which protects the DNA, as the following processes are meant to break apart the cell walls of the E. coli bacteria. Now we place the 384 well plate on the thermocycler. The thermocycler very precisely heats and cools each plate repeatedly. At this point, our plasmid with the DNA insert is still contained within the E. coli bacteria. We separate the plasmids from the E. coli by heating the plate to 95 degrees Celsius. That's almost the boiling point of water. This is just enough to burst the cell walls of the bacteria, but not damage the DNA of the plasmids. Now each well of the plate has a solution of plasmids and broken bits of E. coli bacteria. The next step is to duplicate or amplify the plasmids. A process called rolling circle amplification is used. To start the rolling circle amplification, we add a Templify reagent to the solution in the wells. The Templify reagent consists of dNTPs, which are synthesized free nucleotides, the enzyme that builds the duplicate strands of plasmid DNA, and random hexamer primers, which give the Phe29 polymerase a starting point on the plasmid to start the replication process. The process of rolling circle amplification begins when the random hexamer primers locate and bind to complementary sequences on the plasmid template DNA. The prime DNA is replicated. The enzyme continues to replicate the template DNA until it encounters a double-stranded region. but the enzyme used for rolling circle amplification displaces the double-stranded region, unzipping the DNA as it replicates the template DNA. The unzipped DNA can now be primed and replicated. Because the hexamer primers have all possible ATGC letter combinations, multiple locations are primed. The result is the snowflake-like structures that characterize the rolling circle amplification process. Now we allow this solution to incubate for another 18 hours at 30 degrees Celsius. We do this by placing it in an incubator. For the last 18 hours, only the plasmids have been replicating in each well of this plate. Now we are ready for the actual sequencing part. When we sequence DNA, we want to determine the identity of each nucleotide and the order. We explained that we can tell the identity of each nucleotide in the sequence by using a fluorescent terminating nucleotide. We determine the order by building many copies of different portions of the target DNA, all from the same start point. We then line up all the different lengths of replicated DNA segments from shortest to longest and read the terminating nucleotide by shooting it with a laser to make it fluoresce. But how do we determine where the copying begins? What is preventing us from copying a portion of the plasmid or the E. coli DNA, which is also floating around in the same solution? Well, if you remember, PUC18 plasmid and E. coli have known genomic codes. Also, the sheared segments of DNA that we want to sequence were inserted in a very specific location in the plasmid DNA. The breaking point where we inserted the sheared segment of DNA into the plasmid was in the LAC-Z gene that turned colonies of E. coli bacteria blue. So we know the sequence of nucleotides before and after the segment of sheared DNA that we inserted. In order to start the replication process, we need primers that give the polymerase a place to start replicating the DNA sequence. We chose primers that fit immediately to the left or right of the area where the inserted fragments sit. That way we know exactly where the starting point of the replication begins. Okay, so back to our 384 well plates full of plasmids. We know that we can start copying the target segments of DNA inserted into the plasmid, either forward or backward along the plasmid, depending on which primer we use. However, we can't sequence the DNA if we start replicating segments of DNA both forward and backward in the same well. So we take the solution from each well and transfer it to two new 384 well plates using robots. We then add forward primers to one set and reverse primers to the other set. Next, we add more free nucleotides, Taq polymerase to assemble the new strands, and the fluorescently marked nucleotides. This then goes on to a thermocycler. The thermocycler repeatedly heats and cools the solution to start and stop the segment replication process. Let's take a look at how this works. At the beginning of the cycle, the solution is heated to 95 degrees Celsius. This causes the DNA to unzip and separate into two single strands of DNA. The temperature is then lowered to 50 degrees Celsius. At this point, the primers settle onto the single strands of the DNA. The temperature is then raised to 60 degrees Celsius. At this temperature, the Taq polymerase is activated and starts assembling dNTPs along the single strand of DNA until it randomly incorporates a fluorescently marked nucleotide, or ddNTP. At this point, the chain is stopped. The temperature is then raised again to 95 degrees Celsius, and the segment that had just been made separates from the plasmid DNA. The temperature is then lowered to 50 degrees Celsius, and a new primer settles on the plasmid DNA, and the cycle repeats. The reactions are cycled from 20 to 99 times. The millions of segments this process creates, each with fluorescent terminating nucleotides, are called Sanger fragments because they are the result of the Sanger method of sequencing.